Okay, hey guys. So if you hear sound in the background, it's probably the dishwasher because I'm in the dining room. Okay, but I kind of just wanted to be able to break down the practice questions for you guys in the genetics lecture. Okay, so it's gonna be weird because I can't physically teach you, so I'm gonna be like reading through and writing stuff down and then showing you on the screen. Okay, so the first one is on slide 18. If you're going through the slides, I know some of y'all were doing it with the videos. So Mendel found that crossing wrinkle-seeded plants with pure round-seeded plants produced only round-seeded plants. So if you cross the two and there's only producing one, you know that that one that is produced has to be dominant. So we're just going to do capital R for round seeded. Okay. And then we'll do lowercase r for wrinkled. Okay. So that's all I have. So they want to know what genotypic and phenotypic ratios can be expected from a cross of a wrinkle-seeded plant and a plant heterozygous for this trait. Okay, so they want to know, or they're saying that a wrinkle-seeded plant, so if it's wrinkle-seeded, that means it has to be homozygous recessive. So it's a cross between a little r and a heterozygous, so a capital R with a lowercase r. Okay, so that would be the two genotype crosses. Now they want to know the ratios that can be expected. So that means their offspring. So we will make a Punnett square. Okay, so I have capital R, little r, little r, little r. This is how I set up my Punnett square. Okay, so we have the heterozygous plant at the top and then the homozygous recessive on the side. It doesn't matter where you do it, who's on top or who's on the side. So then we'll just cross them. So then for the ratios, we're going to get, we have 50% that are going to be heterozygous for round seeds. And then we have 50% that are going to be homozygous for wrinkled. Okay. So, yeah, so that's what they're asking for the ratios. Okay, so they may ask you for the ratios in terms of like percentages, or they could ask for like the 2-to-1 or 1-to-2 or 2-to-3-to-2, whatever. So this is, a 2-to-2 or 1-to-1. Okay, so that's the first part. Now the second question on that slide, yellow pods are dominant to green pods in peas. So we're going to do now a capital Y for yellow and a lowercase y for green. What is the probability that the second generation of plants will have yellow pods if the parental generation is homozygous dominant and homozygous recessive for the pod trait? So they want to know, sorry guys, I need more of this. Okay, so they want to know the second generation, not the first, not F1. Okay, so first we have to do the cross of the parents to get F1. Okay, and they just want to know the probability. So we're crossing a homozygous dominant, so capital Y. I'm going to do another Punnett square. Capital Y. And then homozygous recessive, so lowercase y, fill it out. So filling it out, that means F1 generation is going to be completely heterozygous dominant for the trait. Okay, so F1 generation is 100% heterozygous dominant, which means they are all going to be yellow. Okay, so they are all going to be yellow. So now they want to know, sorry, I'm like reading underneath. If the parental what is the probability that the second generation of plants will have yellow pods? Okay, so if all of them are going to be like this, you would basically do a Punnett square comparing two of them together. Since it's 100%, it's pretty easy. We just have our second Punnett square for the F2 generation is going to be our heterozygous versus our heterozygous. Okay, so then fill it out. So then that means 75% of F2 is going to be yellow. Okay, because there's only one that is homozygous recessive. Okay, so all the rest have a capital Y, so 75% are going to be yellow. Okay, so it can be homozygous dominant or heterozygous dominant. So those are those first two problems on slide 18. We go through to find the next one. Okay, so the next one is on slide 24 if you're doing the slides. I know most of us are doing the video. Okay, hold on, Baron Oriole come in. Chaos. Okay, so for this next problem, in dogs, there is a hereditary deafness caused by a recessive gene. A kennel owner has a male dog that she wants to use for breeding purposes if possible. The dog owner wants to know if the male dog is a carrier for deafness. So the owner mates the dog to a deaf female. The ensuing litter contains 3 hearing dogs and 1 deaf dog. What are the genotypes of both parents? So they're saying that the hereditary deafness is caused by a recessive gene. So if they have the deafness, that means that they have to be homozygous recessive. Okay, so let's just use the letter D for deafness. So it would be lowercase d if you are deaf. Okay. Let's see. Kennel owner has a male dog that she wants to use for breeding purposes if possible. The dog owner wants to know if the male dog is a carrier for the deafness. Owner mates the dog to a deaf female. The ensuing litter contains 3 hearing dogs and 1 deaf dog. So we are doing a cross between a deaf dog. Okay, so I'm creating my Punnett square with our homozygous recessive for 1 deaf dog. Okay, and that's the female. And then the male, but they want to know the genotypes of the parents. So obviously we know one of the genotypes of the female. We just have to figure out the male if the litter contains 3 hearing dogs and 1 deaf dog. Well, okay, so if we're going to do where about 75% of the litter has 3 hearing dogs, we know that we need that the male is not deaf, so it's not going to have the homozygous recessive as its genotype. So if we're wanting a 75% chance, the best option is to, have that dominant trait usually be homozygous dominant, because if you want it to match to where it's going to be the dominant trait over the recessive and you only have 1 recessive showing up, you have to kind of, I guess, play with whether the male is now going to be heterozygous dominant or homozygous dominant. Okay, so let's try it with heterozygous. Okay. Okay, so that's what we get if the dad is heterozygous dominant. Okay, so now let's try it with homozygous dominant. Okay, so now we did the homozygous dominant versus homozygous recessive. Okay, so obviously, you know, not all litters of dogs are going to just have 4 puppies, right? So the best option for this to have the genotypes would be, since you can get the dominant and the recessive, okay, it's going to be your homozygous recessive female with your heterozygous dominant male. So that way you can end up getting at least 1 deaf dog. Okay, so that's that problem. Let's move on to the next one. Okay, so it's slide 29 if you're doing the slides. So in mice, the ability to run normally is a dominant trait. Okay, so we have normal running as a dominant trait. Mice with this trait are called running mice. The recessive trait causes mice to run in circles only. Okay, so mice with this trait are called waltzing mice. Hair color is also inherited in mice. Black hair is dominant over brown hair. Determine the genotypic and phenotypic ratios of a cross of a homozygous running homozygous black mouse with a heterozygous running brown mouse. Okay, so this is a dihybrid cross. Okay, so this Punnett square is 16 boxes. It's going to be a lot, but it's okay because I'm here. Okay, so let's write out our genotypes first. So we can see that running is dominant. Okay, so I'm going to put capital R for running and lowercase r for waltzing. And then we have capital B for black hair, which is dominant, and lowercase b for brown hair, which is recessive. So now we are crossing. This is what I have so far. Okay, so now we are crossing a homozygous running. So capital R with a homozygous black. Okay, we're crossing that mouse. Okay, so we have the homozygous running and then homozygous black. We are crossing that with a heterozygous running, so capital R, lowercase r, and a brown mouse. Okay, so those are the 2 parents. So now we have to find the genotypic and phenotypic ratios of their offspring. Okay, so those are the parents. So now we're going to draw a 16 box Punnett square. Okay, so I have my 16 box Punnett square, and I started kind of getting ahead to write the genotypes of what's going to go on the tops and on the sides. So the way that we're going to do this is, I don't know if we remember or if we've heard of the FOIL method in math. Okay, so the FOIL method stands for first, outer, inner, last. Okay, so what we're going to do is we're going to put the first letter of the genotype, okay, for running, with the first letter of the genotype for the hair, okay, for black. So first, So that's how we have our first genotype here, capital R, capital B. Yes, obviously all of these are going to be capital R, capital B, but we can see for the second one how it's going to change. Okay, so now we're going to do outer. So the outermost letters, then the innermost letters, and then the last letters. Okay, so you'll just put those together like that. Okay, so if you need more help in understanding the FOIL method, then just let me know. So then I can kind of show you in another way if necessary. So we have to do first, outer, inner, and last of the second one. So your first one should be capital R, lowercase b. Outer is going to be capital R, lowercase b. Inner is lowercase r, lowercase b. And last is going to be lowercase r, lowercase b. So this is what our Punnett square is going to look like. Okay. So now we have to combine these. So what we're wanting to do is to keep all of the same letters together. So we want to keep our R's together and keep our B's together. But capital always comes before lowercase. But if R, like let's say we had two lowercase r's, okay, that R is going to come before the B. You only keep the capital first in the actual trait itself. So just for running, the capital R would always come before the lowercase r, and just for hair, the capital B would always come before the lowercase b. Okay? So then we're going to write out and just match. Okay? So we just write out all of the genotype there. Okay? And so that's one of the possible genotypes for our mice. Okay? So then we're going to go through, fill all the rest out. So my filled out Punnett square should look like this, okay? So now we have to find out the genotypic and phenotypic ratios, okay? So basically we just need to count how many of each. Okay, so I just counted up and wrote on the side the different genotypes that I see, okay? So obviously there's only two genotypes, is 50-50. For those genotypes, and then 100% of the offspring are going to be running mice with black hair. Okay, so this one was pretty easy in terms of the ratios. Yes, it can get way more complicated, but these dihybrid Punnett squares are the most complicated Punnett squares. So if you need help with them, please reach out to me so I can give you some more practice, maybe like one-on-one. Because it can get pretty chaotic with counting the different genotypes. Okay, so let's move on to the next one. Okay, so the next one is the very next slide, on slide 30. And they're saying you are a world famous breeder of purple people eaters. Tragically, your entire stock perishes in a fire except for two individuals. The two individuals are both heterozygous for all four traits desired. The four traits are as follows. 2 eyes is dominant to 1 eye. 2 horns is dominant to 1 horn. Not flying is dominant to flying. And purple is dominant to white. Okay. The traits that bring the big bucks are 1-eyed, 1 horn, flying, purple people eaters. If you were breed the remaining 2 PPEs, what are the odds that the offspring will have all of the traits that you will be able to sell for those big bucks? So this one is way, way more complicated, okay? Because we're looking at 4 different traits. Okay, so your AP exam is not going to give you a problem with 4 different traits. Okay, if they do in those 2 FRQs, you can slap me. They're mainly just going to be focusing on dihybrid crosses, okay? So you don't have to worry about it too much. So we're going to skip that one. Okay, so the next one is on slide 37. Saying a wealthy elderly couple died together in an accident. Soon a man shows up to claim their fortune, saying that he is their only son who ran away from home as a boy. Other relatives dispute his claim. Hospital records show that the deceased couple were type A, B, and O, respectively. The claim to the fortune was type O. Could he be their son? Use a Punnett square to explain. So the best way to go about this is to go back to the previous table in the PowerPoint to see what the genotypes for the different blood types are. Okay, so once you do that, you would see that type AB blood, the genotype is capital I A, capital I B, and then the genotype for type O is just lowercase I, lowercase I. Okay, so you would see that the genotypes are like this. Okay, so that's the genotypes for the deceased couple, and the son is claiming that he was type O. So we need to use a Punnett square. It's going to be a dihybrid Punnett square to see, oh no, it's not, sorry, to see if it's a monohybrid Punnett square to see if he could be their son. So as you can see, he could not be their son, okay, because their offspring could not have type O, okay? They would have type A or type B blood only, okay? There's that one. Okay, the next one is on slide 38. So in some chickens, the gene for feather color is controlled by codominance. The allele for black is B and the allele for white is W. The heterozygous phenotype is known as erminate. If 2 erminate chickens were crossed, what are the genotypic and phenotypic ratio probabilities of the offspring? Okay, so when we go over codominance, that's when we have one that neither of the traits really outdo the others, so both can be present. So as you can see in that picture, you can see the chicken is like mixed. It's kind of like speckled looking with black and white. Oh my god, it's like Oreo. Okay, so they're saying that 2 erminate chickens were crossed. What are the genotypic and phenotypic probabilities of the offspring? So we're saying that B is black and W is white. Okay. And it's a heterozygous phenotype. So then that would mean if 2 of them were crossed, so it's a B, B, W. Okay, so that's what you would be crossing because they're saying it's a heterozygous phenotype. Okay, so we're going to do a dihybrid Punnett square for this to see. Okay, so I have my Punnett square 16 boxes, so now I have to do the FOIL method for first, outer, inner, last, okay, to get the genotypes that are going to be on the top and on the side. Okay, so since they are both heterozygous, the genotypes on the top and the side are going to be the same, so we just have to match them up, okay? So you'll match up the 4 and see what you get. Okay, so this should be the filled out Punnett square, okay, with what you get. And they want to know what are the genotypic and phenotypic probabilities of the offspring. Okay, so now we're going to kind of count out all the ones that are present for the genotype and see what those phenotypes mean. Okay, so I wrote out all of the genotypes that I saw and counted how many of each there were. And this is what I got. So obviously the greatest number is going to be those that are the erminate, sorry. So they are heterozygous dominant for that trait of codominance, okay? Those had 4 for the offspring. Now to make sure that you have the right number, you want to add them all together, okay? So 1 plus 2, 3, 3 plus 2 is 5, 5 plus 1 is 6, 6 plus 4 is 10, 10 plus 2 is 12, 12 plus 3 is 15. So Miss Taylor did not add up correctly. Never mind, I was right. I just can't count correctly and add correctly because this is only cup number 2. So my B. Okay, so you have your numbers here, so then you're able to get the different probabilities of the different offspring, so you can see who would be, you know, black dominant, white dominant, or the erminate mix. Okay, so now we'll move on to the next problem. Don't know if there is another one. Okay, so that is it from what I can see. If I missed anything, let me know. If you guys needed more help and didn't understand, let me know also. But hopefully this helped. Okay, bye guys.